Let's tie it all together. How does asymmetric cell division and induction work together to drive development? Consider how asymmetric division initiates differences between cells and how those cells then influence each other's fate through induction. This interplay is essential for creating the body's complex architecture from a single fertilized egg. Now we'll lastly turn our attention to the placenta, a remarkable but temporary organ that's formed from both embryonic and maternal tissues. It serves multiple roles: nutrient exchange, waste removal, gas exchange, and endocrine signaling. Understanding placental development is crucial not only for obstetrics, but also for recognizing complications like preeclampsia or intrauterine growth restrictions. Here are some review questions to go through. Things to consider: which embryonic cells form the placenta, which part of the placenta comes from the embryo and which from the mother, which vessels carry oxygenated blood in fetal circulation, and reflect on the four major circulatory adaptations: the ductus venosus, foramen ovale, ductus arteriosus, and the unique role of umbilical vessels. These adaptations are crucial for fetal survival in a low oxygen environment. During implantation, the trophoblast will continue to differentiate, and it will form these two layers: the cytotrophoblast and the syncytiotrophoblast. The cytotrophoblast remains cellular, but the syncytiotrophoblast will become multinucleated and the invasive layer. These cells break down maternal tissue and establish the foundation for placental blood flow. The embryo contributes to the placenta via the chorion. This develops from the trophoblast and mesoderm. Blood vessels develop within the chorion and connect to the umbilical artery and vein. This allows for maternal fetal exchange of blood and supplies, essentially. Importantly, the fetal and maternal blood supplies typically do not mix directly, although some fetal cells can enter into maternal circulation. We can walk through this process here on this slide, looking first at our implanting 8-day blastocyst, and we'll zoom in. The syncytiotrophoblast will erode the endometrium as seen here. The embryonic disc is now separated from the amnion by a fluid-filled space. So we have this bilayered embryonic disc with our epiblast here surrounding the amniotic cavity and our hypoblast here. And below our hypoblast, we see kind of the yolk sac down below. If we go to day 12, implantation is now complete. We can see that we have cells that have covered this developing embryo. Extra embryonic mesoderm is forming in a discrete layer beneath the cytotrophoblast. So we have our bilayered embryonic disc here. So we have the epiblast and the hypoblast layer. We have our amniotic cavity. We have the amnion. We have our cytotrophoblast. We have our yolk sac. We have our extra embryonic mesoderm that's formed. And we have the formation of a chorion, as mentioned. By the 16-day embryo, the trophoblast and associated mesoderm have become the chorion. So we will actually have the formation of this chorion here. The chorionic villi are forming these kind of extensions or projections. And the embryo exhibits all three germ layers. So we have all three layers: ectoderm, mesoderm, and endoderm. We also have a yolk sac and an allantois. The placenta is fully formed by the end of the first trimester, which is important to note. From that point on, it manages all major exchange functions: oxygen, nutrients, waste, and hormones. The placental barrier will consist of fetal capillary endothelium and chorionic membranes. While fetal and maternal blood typically do not mix, this barrier allows for efficient diffusion and is selectively permeable. The embryonic portion of the placenta will arise from the inner cell mass and the extra embryonic mesoderm. These will form the chorion, which develops chorionic villi. We can see some of these in that previous picture and some kind of here, although it's challenging to see. But the chorionic villi are finger-like projections that extend into paternal tissue. These villi are filled with fetal blood vessels and lie in maternal blood-filled spaces, which maximizes exchange while keeping the circulations separate. The chorionic villi will penetrate into large maternal blood-filled lacunae, which are established by the syncytial trophoblast. The villi will bring fetal capillaries into close proximity with maternal blood, which the setup enables efficient exchange of gas and nutrients, as well as efficient removal of wastes. Structurally, this is where the maternal and fetal physiology most closely interact. The maternal contribution to the placenta is called the decidua basalis. This is derived from the endometrial stratum functionalis. As the fetus grows, the decidua capsularis thins and degenerates, while the decidua basalis becomes the main maternal support structure. The combined structure of chorionic villi and decidual tissue forms the mature placenta. Beyond exchange, the placenta is also a powerful endocrine organ. It has endocrine function, as early on it secretes HCG to maintain the corpus luteum. Later, it will produce estrogens and progesterone, which will support pregnancy and prepare the breasts for lactation. It also will relax the human placental, or release the human placental lactogen, relaxin, and chorionic thyrotropin, these are hormones that the maternal body helps or uses to adapt to pregnancy and also uses to support fetal development. So that brings us to the end of this lecture little series. I hope that now you have a clearer understanding of how we go from a single fertilized cell to a complex organized embryo supported by the placenta. If you have any questions, feel free to reach out and review any areas you'd like to revisit prior to moving on to the next topic.